Okay, I'm Haylea Mannebach, and this is my name, and today's date is September 16th. And I did my presentation on the different types of sensory receptors. So before we get into all the different types of sensory receptors and their connection to the central nervous system, I want to first just break down the three different types of sensory neurons you might see in the nervous system. The first being a simple receptor. They have free nerve endings. They may or may not be myelinated, and they're really kind of more exposed to elements. They don't have a lot of shielding. They don't have a lot of myelination. And the second one, the complex neural receptors, these are enclosed by connective tissue. This start of where it's sensing the stimulus is enclosed by that connective tissue, and this is an example of the thickening corpuscle, which is found in the skin, and it senses pressure. And the third one is actually non-neural receptors. And they're for the most specialized senses. This is actually a hair cell found in the ear, and for these receptors, they sense a stimulus and they release a chemical to the associated neuron that then sends the message to the central nervous system. Okay, so for the main classification for the four different types of sensory receptors, sensory receptors are classified based off of the type of stimulus that they can detect. So the first one being a mechanoreceptor. The mechanoreceptors sense mechanical changes, so things like pressure, stretching in the cell, vibrations, acceleration, sound, anything that's mechanically based. And mechanoreceptors are really important for touch, hearing, balance, blood pressure, different things like that. Chemoreceptors sense chemicals, so this could be something as simple as your blood gases, so oxygen or CO2 in your blood, your pH in several different scenarios for organic molecules like glucose or taste or smell. So chemoreceptors are really important for the senses of smell and taste. Photoreceptors sense photons of light. So they're really important for vision. And the last one, thermoreceptors, sense temperature, changes in temperature. So you might find them again in those free nerve endings that we talked about in the first slide. Thermoreceptors are going to be under those sections. Really important for touch, but also thermoreceptors are found in the internal organs like the liver, the skeletal muscle, to regulate the internal temperatures, temperature, and homeostasis. So for each of these receptors, you need an adequate stimulus, meaning that each receptor has only certain stimulus that they preferentially detect. And so no matter what the stimulus is, the response for each of the neurons is pretty is alike a lot of the times. So they need to reach the stimulus needs to reach a certain threshold in order to activate the cell. Once that cell is activated, normally either ion channels open or close. The most often case we see is the sodium ion channel opens and causes an influx of sodium to enter the cell, depolarizing the interior of the cell, causing it to be more of a less negative, more of a positive charge on the inside of the cell. And if that activation were to reach a certain threshold, here is seen as negative 55 millivolts. If it reaches that certain threshold, then a depolarization of the entire cell would ensue. And that's how it would actually send its signal to the central nervous system from the peripheral. But it has to reach that threshold in order for the signal to be sent. Any graded potentials or the change in the sensory cell membrane are known as receptor potentials. So this is the reaction that any neuron, no matter the stimulus or the type of sensory receptor it has, a lot of them have this type of reaction to a stimulus for an actual potential to be created. Not all of them, but a lot of them. So there's something else that's important to take note of is that in all somatic, sensory, and visual neurons, there's something called a receptive field. So this is the area maybe on the skin, if it's like a mechanoreceptor or thermoreceptor, or in your visual sense, it's the place that you can see. So the receptive fields are where you're sensing whatever the stimulus is, and then these primary sensory neurons are again those neurons that are sensing whatever the stimulus is. And if these primary sensory neurons converge into a single secondary sensory neuron, once that convergence happens, then the stimulus that's perceived gets all integrated into one secondary sensory neuron, so the central nervous system isn't able to differentiate apart these two different points here, because the receptive fields are larger and they're overlapping. So this is why, like, if you did a 2-point test on like your legs or your arms, you won't be able to differentiate between the 2 points. But if the receptive fields are smaller and the primary sensory neurons have a 1-to-1 ratio with one primary sensory neuron connecting to a single secondary sensory neuron, then the receptive fields are much more sensitive, and your brain would be able to detect between the 2 different points given the 2 different stimulus. So lastly, now that we've kind of talked about all the different types of receptors, it's important to ask, how does the central nervous system distinguish between the different stimuli if all of what the brain is receiving from the peripheral nervous system are just a whole bunch of action potentials that essentially look the same, and that the cell is being depolarized as it travels towards the central nervous system. So the central nervous system is able to differentiate these the different stimuli based off of 4 criteria. The first being the sensory modality, or basically the nature of the sensor. Like we talked about earlier, each sensor is receptive or really sensitive to their specific type of stimuli, and each then sensory pathway goes to a specific portion of the brain. So those photoreceptors in the eye detecting vision go to the visual cortex. The somatic senses and the peripheral nervous system, so like hands, legs, things like that, they are going to go to the primary somatic sensory cortex. Those mechanoreceptors in the ears sensing sound are going to go to the auditory cortex. The chemoreceptors in the nose sensing smell are going to go to olfactory cortex. So all of these different neural pathways have a very specific portion of the brain that they terminate at that tells the brain what type of sense it originated from. And that same thing kind of goes with the location of the stimulus. Let's say a touch neuron or a touch sensory neuron in your left hand is going to go to a very specific place in the somatic sensory cortex, saying that it's coming from your left hand. So where it terminates in the brain tells you what type of sense it is, and what body part it's also coming from. Another thing that helps the brain differentiate between the senses is the intensity of the stimulus, and that can be seen from the frequency and what we call a population coding. So actually how many receptors are being activated tells the more sensors that are activated, the more intense the stimulus, and then the frequency of the action potentials being sent down the neuron. The more action potentials that are being sent down the neuron, the more intense the stimulus is. And then the last one is the duration of the stimulus. So actually the duration of how the action potential is being sent down the neuron as they go. However long they're lasting will tell us the duration of how long the stimulus is lasting. There are 2 different types of receptors that you might have in this case, but if you have a tonic receptor, no matter how long the duration of the stimulus, your neurons are always going to be sensitive to those action potentials, and you're always going to be sensing whatever that stimulus is. For basic receptors, you after a certain amount of time, your neurons are going to be no longer sensitive to that stimulus, and you're no longer going to have an action potential created, so you're no longer going to be sensitive to that stimulus. So that's seen in smell. So once you're exposed to a smell for a certain amount of time, you're no longer going to be sensitive to it. Same thing for like why we can wear our clothes and our pants every day without feeling them. Once your legs and your arms, those touch receptors on the surface, are exposed to the closed for a certain amount of time, we no longer are sensitive to them. So that's my presentation about the different types of stimulus and the different types of sensory receptors. I'm also going to turn in the study aid that will help. And so, enjoy.